welcome to Chapter 7, Annotation and Databases for Genetic Variants. This chapter addresses what is, in my view, the most practically challenging and intellectually fascinating problem in modern clinical genomics, not generating the data, which we now do routinely, but interpreting it. When a whole exome or whole genome sequencing experiment is performed on a patient, the output is not a diagnosis. The output is a list of genomic positions where the patient's DNA differs from the reference genome. A typical exome generates roughly 20,000 such differences. A typical whole genome generates 4 to 5 million. The overwhelming majority of these differences are completely harmless common polymorphisms that make each person genetically unique. A small number, potentially just 1 or 2, may be the cause of the patient's disease. The challenge is finding that needle in the haystack, and that challenge requires a systematic set of tools and databases for adding meaning to each variant. This process is called variant annotation, and it involves answering 4 fundamental questions for every variant. Where in the genome is it located? What functional consequence does it have? How common is it in the general population? And is it known to be associated with any disease? We have built remarkably powerful resources to answer each of these questions, and this chapter will give you a thorough understanding of those resources, how to use them, and their limitations. By the end of this lecture, you will be able to trace a variant through the complete interpretation pipeline. Let me begin by making the scale of the interpretation challenge concrete. When you sequence someone's genome using next-generation sequencing and compare the results to the reference genome, here's what you find. Approximately 4 to 5 million single nucleotide variants, positions where the patient has a different base than the reference. These arise because the reference genome represents one particular set of alleles, and every individual differs from it at millions of positions. The majority of these variants are common in the human population. They are not mutations, they are normal genetic variation, the differences that make each of us genetically unique. Additionally, approximately 400,000 to 500,000 small insertions and deletions called indels, where the patient's genome has bases inserted or deleted relative to the reference, and thousands of larger structural variants, deletions, duplications, inversions, and translocations involving hundreds to thousands or more base pairs. Now, here is the core problem of these 4 to 5 million total variants in a typical genome. The number responsible for a specific Mendelian genetic disease in a particular patient is usually exactly 1, sometimes 2 for recessive disorders. The signal-to-noise ratio is roughly 1 in 4 million. Finding that one causal variant among millions of benign ones is the fundamental challenge of clinical genomics, and it is a challenge that cannot be solved by looking at each variant manually. It requires systematic, automated annotation comparison to population. Variant annotation is the process of systematically adding biological and clinical context to each variant identified in a sequencing experiment. For every difference between the patient's genome and the reference, annotation answers 4 fundamental questions. The first question is location. Where precisely in the genome does this variant fall? Is it within a gene or between genes? If within a gene, is it in an exon, an intron, the promoter, or the untranslated regions? If in an exon, which codon does it affect? Knowing the location tells you immediately whether the variant has the potential to affect protein function or gene expression. The second question is function. What does this variant do to the gene product? For a coding variant, does it change an amino acid? Does it create a premature stop codon? Does it disrupt a splice site? A synonymous change that swaps one codon for another, encoding the same amino acid, is much less likely to cause disease than a nonsense change that truncates the protein. The third question is population frequency. How common is this variant in the general human population? This is a critical filter because variants that cause severe early onset genetic diseases are kept at very low frequency by natural selection. A variant present at 1% frequency in the population cannot be the single cause of a rare disorder that affects 1 in 10,000 people. Population frequency data therefore allows immediate filtering of the vast majority of benign common variants. The fourth question is clinical significance. Has this specific... This is the most direct evidence for pathogenicity. and comes from databases of clinically documented variants with expert curation. Three major software tools automate the first two annotation questions, namely location and functional consequence, for every variant in a VCF file. Each has distinct strengths, and in practice, many analyses use two or three in combination for cross-validation. The first is VAP, the Variant Effects Predictor, developed and maintained by the Ensemble project at the European Bioinformatics Institute. VAP excels at comprehensive location mapping. For every variant, VAP looks up its precise genomic coordinates in the Ensemble annotation database and reports which gene it falls in, which transcript, which exon or intron, and what protein change results if it is a coding variant. VAP is particularly valuable for whole genome analysis because it annotates non-coding variants against Ensemble's catalog of regulatory elements, flagging variants in promoters, enhancers, and other functional non-coding regions. The output for a pathogenic CFTR variant might read CFTR exon 11 stop gain pGlyph42*, indicating that the variant creates a premature stop codon at position 542 of the CFTR protein. The second tool is ANNOVAR, which stands for Annotate Variation. ANNOVAR's strength is database integration. Rather than focusing primarily on location and consequence, ANNOVAR retrieves information from dozens of external database integration. ANNOVAR's strength is database integration rather than focusing primarily on location. The third tool is SNPF, which classifies variants by their predicted impact on a four-tier scale: high impact for stop gains, frame shift mutations, and splice site disruption; moderate impact for missense variants that change the amino acid; low impact for synonymous variants that do not change the amino acid; and modifier impact for non-coding variants. This impact classification system provides rapid triage for large variant datasets. Now let us turn to the databases themselves, starting with the most foundational layer of the three-tier variant database system. The first tier is DBSNP, the single nucleotide polymorphism database, which functions as the comprehensive catalog of documented human genetic variation. DBSNP is maintained by the National Center for Biotechnology Information and has been accumulating variant records since 1998. It currently contains over 1.1 billion documented variant positions. Despite its historical name, which reflects that single nucleotide polymorphisms were the dominant focus when the database was founded, DBSNP now catalogs not just SNPs, but also small insertions and deletions, multinucleotide variants, and other classes of small variants. The core feature of DBSNP is the RS number, which stands for Reference SNP. Every variant documented in DBSNP receives a unique RS number that serves as a stable identifier for that specific genomic change. When a clinician reports that a patient carries RS 429358, they are referring to a specific well-documented variant in the polyprotein E gene that is strongly associated with Alzheimer's disease risk. The RS number allows unambiguous communication about specific variants across different publications, databases, and clinical reports. The practical value of DBSNP in variant interpretation is primarily as a filter. When a variant at 5% frequency in a large population survey cannot be the sole cause of a condition affecting 1 in 10,000 people. This population genetics argument, applied systematically through DBSNP lookup, filters out the vast majority of the millions of variants in a genome and focuses attention on the rare ones with the highest prior probability of pathogenicity. The second tier of the variant database system is GNOMAD, the genome aggregation database, which is the most powerful tool available for assessing how common a variant is across diverse human populations. GNOMAD was developed at the Broad Institute and represents a quantum leap in the scale and quality of population frequency data available to variant interpreters. GNOMAD aggregates high-quality sequencing data from over 140,000 individuals spanning multiple ancestry groups. These individuals were sequenced as part of various large-scale research projects and were specifically selected to exclude individuals with rare pediatric diseases that might be caused by the variants being studied. The result is a reference database reflecting the normal range of genetic variation in apparently healthy adults. For every position in the human genome, NeoMed reports how many of its 140,000 individuals carry the variant and what the allele frequency is overall and broken down by ancestry group. These ancestry-specific frequencies are crucial. A variant that is absent from European ancestry participants may be common in East Asian or African ancestry participants or vice versa. Using only European reference data to filter variants in a patient of non-European ancestry can lead to both false positive pathogenicity calls for common population-specific variants and false negative misses or rare variants with uncertain population-specific frequency. The key clinical logic of NeoMed is this. If a variant is present at grade, if a variant is completely absent from all 140,000 individuals in NeoMed, that absence is informative. A truly benign variant should appear at some frequency in a database of this size. Its absence raises the prior probability that it has a functional consequence. Let me expand on why population diversity in NeoMed matters so much for clinical interpretation, because this is the point with real consequences for patients from underrepresented ancestries. Genetic variants are not uniformly distributed across all human populations due to the history of human migration, population bottlenecks, and local natural selection. Variants that are rare in one population can be common in another. This population stratification of variant frequencies creates a systematic problem when databases are built primarily from data of European ancestry individuals, as early genetic databases largely were considered a practical scenario. A clinician sees a patient of East Asian ancestry with a suspected genetic heart condition. Whole exome sequencing identifies a missense variant in a cardiomyopathy gene. The clinician checks the frequency in European population databases and finds the variant is absent. This seems suspicious, but if NeoMed's East Asian ancestry subset shows the variant at 2% frequency, the variant is almost certainly benign, a common population-specific polymorphism. Without ancestry-matched population data, this variant would be misclassified as likely pathogenic, leading to an incorrect diagnosis and potentially unnecessary clinical management. The NeoMed data makes another powerful argument available in certain situations, the argument from functional constraint. For some genes that encode the essential proteins, NeoMed data. If a gene encodes a protein where even subtle amino acid changes are poorly tolerated, a novel missense variant in that gene has a higher prior probability of being pathogenic than a missense in a gene that tolerates many common variants. NeoMed provides a metric called PLI, the probability of being loss of function intolerant, that quantifies this gene-level constraint. The third and most clinically actionable tier of the variant database system is ClinVar, maintained by NCBI, where DBSNP catalogs documented variants and NeoMed provides population frequencies. ClinVar provides clinical interpretation. What disease, if any, is this variant known to cause? ClinVar is a database of variants submitted by clinical diagnostic laboratories, research groups, and expert panels, each submission accompanied by evidence and a clinical classification. The classification system uses a 5-tier scale. Pathogenic means the variant is known to cause disease with strong evidence. Likely pathogenic means evidence strongly suggests pathogenicity, but is not yet fully conclusive. Variant of uncertain significance, abbreviated VUS, means there is insufficient evidence to confidently classify the variant as pathogenic or benign. Likely benign and benign complete, the scale at the other end. The clinical workflow enabled by ClinVar is straightforward in principle, though often complicated in practice. A patient presents with a suspected genetic disease. Sequencing identifies a variant in a relevant gene. The clinician queries ClinVar If the variant is classified as pathogenic with multiple lines of supporting evidence from multiple submitting laboratories, and the clinical description matches the patient's phenotype, a diagnosis is confirmed. If the variant is classified as a VUS identified in a patient today may be reclassified as pathogenic or likely benign in 12 to 24 months as more affected individuals are sequenced and more functional data becomes available. For this reason, many clinical genetics programs recommend periodic reanalysis of unsolved cases as databases grow. The variant of uncertain significance, VUS, is one of the most practically important and clinically challenging concepts in modern genetic medicine. Understanding what a VUS is, why it exists, and how to manage it is essential for anyone working in clinical genetics. A VUS is a variant for which there is insufficient evidence to confidently classify it as either pathogenic or benign. This is not a failure of the database or the sequencer. It is an inherent epistemic limitation that arises from the number of possible variants versus the amount of clinical data available. The human genome has approximately 3 billion base pairs. The coding genome alone has roughly 16 million possible positions that could carry a missense variant. Current databases contain clinical evidence for only a small fraction of all possible variants. The vast majority of missense variants are one of a kind, a unique combination of position and amino acid substitution seen in just one or a handful of patients. For such variants, answering the pathogenicity question requires multiple lines of evidence. Is the amino acid position evolutionarily conserved across species? Conservation implies functional importance and raises the probability that any change there is deleterious. Does the variant affect a known functional domain of the protein? Do computational predictors like Revel or alpha missense predict it to be damaging? Is there a family member with the same disease who also carries the variant? This is the VUS problem, and it is particularly burdensome for patients from underrepresented populations where fewer individuals have been sequenced and fewer variants have clinical documentation. Variants common to understudied populations are more likely to receive VUS classifications even when they are benign, simply because there are fewer comparators in the database. For variants not yet documented in ClinVar, computational tools can estimate the likely pathogenicity of a variant based on multiple lines of indirect evidence. These predictors do not replace experimental evidence, but they provide an important additional signal for variant prioritization and interpretation. Three tools deserve particular attention. CAD, the Combined Annotation Dependent Depletion Score, was developed at the University of Washington. CAD takes a fundamentally different approach from tools that look at one feature at a time. It uses a machine learning model trained on millions of variants to integrate over 100 features simultaneously. Sequence conservation across species, protein function predictions, regulatory element overlap, gene expression data, chromatin accessibility, and dozens more. The CAD score is reported on a Phred-like scale. A CAD score above 20 means the variant is predicted to be in the top 1% most deleterious variants across the genome. A score above 30 means the top 0.1%. CAD is widely used as a general-purpose variant impact score applicable to both coding and non-coding variants. REVEAL, which stands for Rare Exome Variant Ensemble Learner, is a machine learning meta predictor specifically optimized for missense variants in the context of Mendelian disease. REVEAL integrates 13 individual missense predictors and outputs a score from 0 to 1, where scores of alpha missense represents the most recent advance applying the structural prediction capabilities of AlphaFold 2 in a variant pathogenicity context. Alpha missense was trained on structural data and evolutionary conservation to predict the pathogenicity of essentially all possible human missense variants, approximately 71 million variants, classifying each as likely benign, ambiguous, or likely pathogenic. Its integration of protein structure information makes it particularly informative for variants in structurally important domains. Before annotation tools can assess where a variant falls in a gene and what its functional consequences. They need a map of the genome that defines exactly where each gene and each exon is located. Two major annotation systems provide this genomic map, and understanding their differences matters for clinical practice. GeneCode is a comprehensive annotation project that provides highly detailed maps of all gene features across the human genome. GeneCode is the annotation system used by Ensemble and by VIP. It defines approximately 45,000 genes in the human genome, including not just the roughly 20,000 protein coding genes, but also thousands of long non-coding RNA genes, pseudogenes, small RNA genes, and other genomic features. GeneCode uses identifiers starting with ENSG for genes and ENSG for transcripts because GeneCode captures the full complexity of the transcriptome and annotates many non-coding functional elements. It is the preferred annotation system for research applications, particularly for whole genome analysis where non-coding variants are relevant, RefSeq. The reference sequence database maintained by NCBI takes a more conservative approach. It focuses primarily on the roughly 20,000 well-validated protein coding genes and provides identifiers in the format NM underscore for messenger RNA transcripts and NP underscore for protein sequences. RefSeq updates less frequently than GeneCode, but its updates are more conservative. When a clinical laboratory reports that a patient carries a variant at NM007294.3:C.5266 dupe, they are using the RefSeq nomenclature that is the agreed upon standard for communicating clinical variant identities. In practice, variant effects should ideally be reported with respect to a canonical transcript for each gene, and discrepancies between gene code and RefSeq canonical transcripts can occasionally affect how a variant's consequence is classified. Annotation databases answer questions about what a variant does biochemically and how common it is in the population. But the most clinically powerful evidence comes from a third type of resource, biobanks, which are large collections of DNA samples linked to comprehensive health records. Biobanks answer the question that databases of healthy individuals cannot. Do people who carry this variant have higher rates of a specific disease? The principle of biobank-based genetic association is straightforward. Recruit large numbers of participants who consent to share their DNA and health data, sequence or genotype their genomes, link the genetic data to longitudinal health records, including diagnosis, medication prescriptions, laboratory values, and imaging results. Then ask, is variant X associated with disease Y? Do carriers of a particular APOE allele have higher rates of Alzheimer's disease? Do people with a specific PCSK9 variant have lower LDL cholesterol and fewer cardiac events? The UK Biobank is the paradigm for this approach. It recruited 500,000 British adults aged 40 to 69 between 2006 and 2010, collected biological samples, and obtained ongoing access to participants' complete National Health Service records. The entire cohort has been whole exome sequenced and is being whole genome sequenced. Studies from the UK Biobank have identified thousands of gene trait associations spanning essentially every. The All of Us program in the United States aims to recruit 1 million participants with a specific focus on ancestry diversity, intending to correct the historical underrepresentation of non-European ancestry individuals in genomic research. By 2024, All of Us had enrolled over 245,000 participants. The importance of population diversity in genomic databases extends beyond the NOMAD's role in filtering common variants. Population-specific biobanks and databases have revealed that the genomic architecture of traits and diseases differs meaningfully across ancestry groups with important clinical consequences. COVA, the Korean variant archive, provides a particularly instructive example. This biobank sequenced 5,305 Korean individuals and characterized the variant frequency specific to the East Asian Korean population. The key finding was that approximately 22.8% of variants identified in Korean patients as potentially pathogenic by European-dominated databases were actually common in the Korean population and therefore benign. This means that using only European reference data would have resulted in almost one quarter more false positive pathogenicity causing Korean patients. The clinical consequences of false positive pathogenic variant cause are serious, unnecessary anxiety, inappropriate prophylactic intervention, incorrect diagnosis, and misdirected treatment. FinGen is a Finnish biobank project with over 500,000 participants. The Finnish population has undergone a series of historical bottlenecks due to geographic isolation and a small founding population. This history created a pattern called the Finnish disease heritage. Rare recessive disorders that are extremely rare elsewhere in the world are enriched in Finland because of the founder effect. Variants causing these conditions reach the elevated. FinGen has been invaluable for characterizing the spectrum of these Finnish enriched variants and for distinguishing rare pathogenic variants from rare but benign population-specific variants. The TOMO project in Japan, the Toku Medical Megabank Organization, has recruited over 150,000 participants, including 3-generation family cohorts, enabling study of how genetic variants interact with environmental exposures over time. Its post-disaster component, following survivors of the 2011 Toku earthquake and tsunami, has added a unique longitudinal dimension. Let me now walk through the integrated clinical workflow that brings all of the annotation tools and databases together to convert a list of millions of variants into a clinical diagnosis. This workflow is the practical implementation of everything we have discussed, and understanding it will help you make sense of real clinical genomics reports. The starting point is the annotated BCF file generated by the bioinformatics pipeline. For a typical whole exome sequencing case, this contains approximately 20,000 variants. The first filter applied is functional consequence. Using SNP-F impact classifications, or consequence term, variants that are synonymous, meaning they do not change the protein sequence, and variants in non-coding regions are typically excluded in a standard coding focused analysis. This reduces the variant count to a few thousand. The second filter is population frequency applied using NUMED data. Variants with a minor allele frequency above 1% in any NUMED ancestry group are excluded as too common to cause a rare Mendelian disease. This single filter applied systematically removes the large majority of remaining variants. In a typical exome, fewer than 200 rare coding variants remain after this filter. The third step is checking ClinVar. Any variant already classified as pathogenic or likely pathogenic in ClinVar that matches the patient's phenotype. Variants are cross-referenced against databases of gene disease association, such as OMIM, the Online Mendelian Inheritance in Man Catalog, to identify which remaining variants fall in genes known to cause diseases resembling the patient's presentation. Variants in unrelated genes are deprioritized. Let me walk through a complete clinical case that illustrates the annotation and interpretation workflow from beginning to end. This case is a composite based on typical presentations of early onset epileptic encephalopathy, and it demonstrates how the tools we have discussed translate to real diagnostic value. The patient is a 4-year-old child with developmental delays, epileptic seizures beginning at 6 months of age, intellectual disability, and abnormal brain MRI findings. Multiple anti-seizure medications have provided incomplete seizure control. The clinical team suspects a genetic etiology. Whole exome sequencing is performed on the patient and both parents. The sequencing generates approximately 25,000 variants in the patient after applying the annotation pipeline, filtering for rare variants affecting protein function. When removing variants common in NOMAD, approximately 200 rare coding variants remain. After checking ClinVar and deprioritizing variants in genes with no known neurological disease association, 40 candidate variants remain. The team then focuses on genes known to cause early onset epileptic encephalopathy. 3 variants fall in genes in this category. The most compelling is the novel missense variant in SCN1A, the gene encoding the sodium channel subunit nav 1.1, this variant was not inherited from either parent. It is absent from all 140,000 individuals. ClinVar contains submissions from 3 independent clinical laboratories classifying similar SCM1 a de novo missense variants as pathogenic in the context of Dravet syndrome. The diagnosis is Dravet syndrome. Treatment is adjusted to avoid sodium channel blocking medications that worsen the condition, and the family is counseled about low recurrence risk given the de novo origin. Throughout the annotation and interpretation workflow, one database sits at the center of gene-to-disease mapping, OMIM, the online Mendelian inheritance in man. OMIM is the authoritative catalog of human genetic disorders and the genes that cause them, and no clinical geneticist can practice without it. OMIM was founded by Victor McCusick at Johns Hopkins University in the 1960s as a printed catalog called Mendelian Inheritance in Man. It has been maintained online since the early 1990s and is now a comprehensive, regularly updated database covering over 27,000 entries for genetic conditions and their causative genes. Each OMIM entry has a unique numeric identifier, preceded by a symbol indicating the inheritance pattern. An asterisk indicates a gene entry. A number sign indicates a phenotype caused by a known molecular basis. An entry beginning with the percent sign indicates a phenotype with a suspected genetic basis, but where the causative gene has not yet been identified. The clinical utility of OMIM in variant interpretation is primarily for phenotype matching. When annotation identifies a novel variant in a previously uncharacterized gene, OMIM lookup will show whether that gene has any documented disease association. Conversely, when the patient has a recognized syndrome, OMIM provides a list of all genes known to cause that syndrome, focusing the variant search on a specific gene set. OMIM is also invaluable for understanding the expected inheritance pattern and penetrance of conditions, which guides interpretation of whether a variant. For autosomal recessive conditions, 2 pathogenic variants, 1 on each chromosome, are required. The American College of Medical Genetics and Genomics and the Association for Molecular Pathology have published a widely adopted framework for classifying the pathogenicity of sequence variants in the context of Mendelian disease. Published in 2015, this framework provides a systematic, evidence-based approach to variant classification that has been adopted by clinical laboratories globally. The ACMG-EMP framework organizes evidence for pathogenicity or benignness into specific criteria, each weighted according to its strength. Evidence is classified as very strong, strong, moderate, or supporting for both pathogenicity and benignness. Criteria are combined using a semi-quantitative scoring approach to reach one of 5 classifications: pathogenic, likely pathogenic, variant of uncertain significance, likely benign. Key pathogenicity criteria include PVS1, a null variant in a gene where loss of function is a known disease mechanism; PS1, a variant at the exact same amino acid position as a known pathogenic variant; PS2, a de novo variant in a patient with a disease confirmed with paternity and maternity testing; PM2, a variant absent from controls in NeoMedi; PP3, a variant predicted to be damaging by multiple computational tools. Key benign criteria include BA1, a variant with allele frequency above 5% in any NeoMedi population; BS1, a variant with a frequency higher than expected for a dominant disorder; BP4. It also makes explicit what evidence is available and what evidence is lacking, which is important for patients who need to understand why their variant is classified as uncertain. In addition to the general purpose databases we have discussed, thousands of locus-specific databases provide highly curated variant information for specific genes or diseases. These specialized resources often contain information not captured in general databases and can be decisive in variant interpretation. Locus-specific databases collect all documented variants in a specific gene along with detailed clinical annotations. For example, the BRR Exchange aggregates variant data for BRR1 and BRR2 from numerous clinical laboratories and research programs worldwide, and expert panels review and classify variants with more granularity than is typically possible for general databases. The LOVD The latent open variation database hosts locus-specific databases for hundreds of individual genes. Expert panel reviews by groups recognize expert panel program represent the highest level of evidence for variant classification. These panels, organized by the Clinical Genome Resource Consortium, bring together disease experts who systematically evaluate all available evidence for variants in their disease area and publish consensus classifications. An expert panel pathogenic classification carries more weight than a single laboratory submission in the ACMGM framework. Expert panels exist for many clinically important genes, including BRR1 and 2, hereditary cardiac channelopathy genes, PTN, TP53, because the same ACMGM evidence criteria can have different weights depending on the gene. ClinGen's gene-specific guidance helps laboratories apply the framework more accurately. One of the most powerful lines of evidence for predicting whether a variant at a specific position is likely to be functionally important comes from evolutionary conservation. If a particular nucleotide or amino acid position has been preserved unchanged across millions of years of evolution in many different species, it is likely because any change at that position is harmful. The logic is as follows. Evolution is constantly testing new variants through mutation. Variants that are neutral, meaning they have no effect on fitness, drift freely in populations and may become common or may disappear by chance. Variants that are harmful, reduce reproductive fitness, are selected against and disappear from the population over time. A position that has been maintained identically in mice, frogs, zebrafish, and flies has been continuously subjected to this selective pressure for hundreds of millions of years and has remained unchanged, which is strong evidence that changes at that position are deleterious. Multiple conservation scoring systems quantify evolutionary constraint at each genomic position. Follow-up scores measure the evolutionary conservation at each individual nucleotide position based on alignment of the human genome to many vertebrate genomes. Positive follow-up scores indicate conservation, negative scores indicate acceleration. GRP, genomic evolutionary rate profiling, similarly measures constraint and nucleotide resolution. For protein coding positions, Blossom scores quantify how often each specific amino acid substitution is observed across. These conservation metrics are incorporated into composite predictors like CAD and Rebel, where they contribute to the overall pathogenicity prediction. Standalone, they are also directly applicable. A missense variant at a position with high phyloconservation conservation score has a higher prior probability of pathogenicity than an equally novel missense at a poorly conserved position. Most of our discussion so far has focused on annotation of single nucleotide variants and small indels, but structural variants, meaning large-scale genomic rearrangements, including deletions, duplications, inversions, and translocations, require a separate annotation framework with their own specialized databases and tools. Structural variants are far less common per genome than single nucleotide variants, but they are responsible for a disproportionate fraction of severe genetic disease. A large deletion removing one or both copies of a critical gene will typically be more damaging than the missense variant in the same gene. Structural variants are also systematically underdetected by short-read whole exome sequencing, as we discussed in the previous chapter, making whole genome sequencing essential for comprehensive structural variant identification. The primary reference database for structural variants is the Database of Genomic Variants, or DGV, which catalogs structural variants observed in healthy control individuals. Like NOMAD for SNPs, DGV allows variant interpreters to assess whether a structural variant is common in the general population. A deletion found in hundreds of healthy individuals is benign. A deletion absent from all healthy controls is suspicious. The ClinVar database also accepts structural variant submissions, and a subset of structural variants are classified with pathogenicity evidence. The DCPA database at the Wellcome Sanger Institute specifically focuses on structural variants in patients with annotation tools for structural variants include Annotation, which systematically annotates structural variants against gene positions, known pathogenic variant databases, population frequency databases, and functional data. A clinically important application of variant annotation that deserves specific attention is pharmacogenomics, the study of how genetic variants affect an individual's response to drugs. Pharmacogenomic variants do not cause disease in the traditional sense. They affect whether a drug works at the intended dose, whether it works at all, or whether it causes serious adverse reactions. The classic example is the CYP2D6 gene, which encodes a cytochrome P450 enzyme responsible for metabolizing approximately 25% of all commonly prescribed medications. CYP2D6 is one of the most polymorphic genes in the human genome, with hundreds of documented variant alleles that affect enzyme activity. Individuals can be classified as poor metabolizers who lack functional CYP2D6 activity, intermediate metabolizers, normal metabolizers, rapid metabolizers, or ultra-rapid metabolizers who have multiple copies of the gene and very high enzyme activity. These metabolizer categories have direct clinical implications. A poor metabolizer given the standard dose of codeine, which requires CYP2D6 to convert it to morphine, will receive no analgesic benefit because the drug is not activated. An ultra-rapid metabolizer given the same dose may experience morphine toxicity because the drug is converted to active metabolite too rapidly. Metabolizer given the same dose may experience morphine toxicity because the drug is converted to active metabolite too rapidly. These are predictable pharmacogenomics. It compiles information on gene-drug relationships with evidence-based assessments and is integrated with the Clinical Pharmacogenomics Implementation Consortium, CPIC, which provides dosing guidelines that can be implemented when pharmacogenomic test results are available as whole genome sequencing becomes more routine in clinical care. Pharmacogenomic data from routine germline sequencing will increasingly be used to guide prescribing decisions. One of the most important concepts in clinical genomics that distinguishes it from most other diagnostic tests is that variant interpretation is not a one-time event. It is a continuous process that should be revisited as databases grow and as new functional evidence becomes available. The databases we have discussed, NOMAD, ClinVar, OMIM, locus-specific databases, are not static. They grow substantially every year as more genomes are sequenced, more patients are studied, and more functional experiments are performed. A variant classified as VUS today, because there is insufficient evidence, may be reclassified as pathogenic in 12 months when additional affected individuals carrying the same variant are reported in ClinVar, or as likely benign when it is found in NOMAD at elevated frequency. These reclassifications have direct clinical consequences for patients who received uncertain results. Studies of VUS reclassification rates have shown that VUS for hereditary cancer genes like BRCA1 and BRCA2 are reclassified at a rate of roughly 2 to 3% per year, and that the majority of reclassifications are toward benign rather than pathogenic. This has important implications for patients who received a VUS result and have been living with uncertainty about their cancer risk. Many clinical genetics programs now offer periodic reanalysis of previously unsolved or uncertain cases. Patients with negative or VUS results are offered. Some research programs use automated systems that flag cases for reanalysis when the new ClinVar submission or anomaly update changes the frequency or classification of a variant found in the patient. The key message is that receiving a VUS result should not be the end of the patient's genomic medicine journey. It should be the beginning of an ongoing monitoring relationship with the genetics team. Let me make the filtering process concrete by tracing exactly what happens numerically when a clinical annotation pipeline processes a typical whole exome sequencing case. Understanding the scale of the reduction at each filtering step helps you appreciate both the power and the potential failure points of the pipeline. Starting point, approximately 25,000 variants identified in a whole exome sequencing run, representing all positions where the patient's coding sequence differs from the reference genome GRCH38. Step 1, remove synonymous variants and non-coding variants using SNPF or BAP impact filtering. After this step, approximately 4,000 to 5,000 variants remain, those with at least moderate predicted impact on the protein sequence. Step 2, remove common variants using nomadic population frequency above 1% in any nomadic ancestry group are classified as too common to cause a rare Mendelian disease and excluded. After this step, typically fewer than 200 rare coding variants remain. This single filter removes roughly 95% of the variants that survived the functional consequence filter. Step 3, apply CleanVar annotation. Any variant with an existing benign or likely benign classification is removed. Any pathogenic or likely pathogenic classification in a gene relevant to the patient's phenotype is flagged as a priority candidate. Remaining unclassified variants proceed to the next step. Step 4, phenotype-driven gene filtering using the patient's clinical features. The analyst, after this step, typically 3 to 10 candidate variants remain. Step 5, computational pathogenicity prediction using CAD, Revel, and AlphaMissense provides quantitative evidence for each remaining candidate. Step 6, inheritance pattern analysis, checking whether each candidate variant fits the expected inheritance mode, autosomal dominant, recessive, or X-linked, given what is known about the candidate gene. The result of this entire funnel is typically 1 to 3 high-confidence candidates that warrant clinical validation. An important but often overlooked tool in the clinical variant interpretation toolkit is RNA sequencing of patient-derived tissue. While DNA sequencing tells you what variants are present, RNA sequencing tells you whether those variants actually affect gene expression or splicing, providing functional evidence that can resolve BUS classification and identify variants that DNA sequencing alone would miss. The rationale for using RNA sequencing in diagnosis is mechanistic. Many pathogenic variants act by disrupting RNA processing rather than by directly changing the protein sequence. Deep intronic variants can create cryptic splice sites, causing the spliceosome to include a segment of intronic sequence. Witronic variants can create cryptic splice sites, causing the spliceosome to include a segment of intronic sequence in the mature messenger RNA. To skip an exome promoter variant can reduce transcription, lowering messenger RNA levels, variants that destabilize the messenger RNA through nonsense-mediated decay can reduce transcript abundance. All of these effects are invisible at the DNA level, but directly visible in RNA sequencing data. In a typical diagnostic RNA sequencing workflow applied to a patient with an unresolved rare disease, RNA is extracted from patient cells or tissue, ideally from a tissue where the candidate gene is normally expressed. If the patient has studies using this combined DNA and RNA approach in patients with rare undiagnosed diseases have shown that RNA sequencing increases the diagnostic yield by approximately 10 to 15% above DNA-based exome or genome sequencing alone. This incremental gain comes primarily from resolving deep intronic variants that create cryptic splice sites and from confirming that suspected pathogenic coding variants actually affect the transcript. I want to introduce a concept that represents the next major evolution in how we perform variant annotation and genomic analysis, the human pangenome reference. This development has important implications for the accuracy of variant interpretation, particularly for individuals from diverse ancestries. The current standard reference genome, GRCH38, and its successor T2T CHM13 represent linear sequences derived from a small number of individuals, predominantly of European ancestry. When we align sequencing reads from a patient to this reference and call variants, we are comparing the patient's genome to a single reference that may not represent the full diversity of human genomic sequences. Regions of the genome that differ substantially from the reference, which are more common in individuals from ancestries not well represented in the reference, are more likely to have alignment errors, incorrect variant calls, and coverage gaps. The Human Pangenome Reference Consortium, which published its first draft pangenome reference in 2023 in the journal Nature, took a fundamentally different approach. Instead of a single linear reference, the pangenome is a graph structure that incorporates the genome sequences of 47 diverse individuals from multiple ancestries and regions of the world. Each node in the graph represents a sequence present in at least one individual, and the edges represent how the sequences connect. A sequencing read can be aligned to the pangenome graph, finding the path through the graph. Segmental duplications and other regions where human populations carry extensive structural variation create systematic alignment errors when using a single linear reference. The pangenome captures this structural variation explicitly, allowing more accurate alignment and variant calling for patients whose genomes carry sequences not well represented in the linear reference. As the pangenome becomes the standard reference framework, it will improve variant interpretation accuracy, specifically for the populations that have historically been most poorly served by genomics. Let me return to a theme we touched on in discussing GEOMAD because it deserves fuller treatment. The systematic underrepresentation of non-European ancestry populations in genomic databases is not merely a scientific limitation. It is a health equity issue with direct consequences for patients. The history of genomic research has been heavily dominated by participants of European ancestry. The reasons are partly historical, reflecting the geography of early genomics research programs, and partly practical, reflecting the locations of well-funded research infrastructure. The consequence is that the variant databases that clinical genetics relies on are biased toward European genetic variation. Variants that are common specifically in African, South Asian, East Asian, Latin American, or indigenous populations are underrepresented in these databases, making variant interpretation systematically less accurate for patients from these ancestries. The practical consequences are serious. Variants common in understudied populations may appear as rare variants in current databases, causing them to be misclassified as potentially pathogenic when they are actually common benign polymorphisms. This leads to false positive pathogenicity codes that are more likely to affect patients of non-European ancestry than patients of European ancestry. Conversely, pathogenic variants enriched in specific populations may be undercharacterized in terms of their disease penetrance and phenotypic, but progress requires active commitment and sustained funding. The scientific case for diverse databases is clear. A more representative reference improves interpretation accuracy for all patients. The ethical case is equally clear. All patients deserve access to accurate genomic interpretation, regardless of their ancestry. These two arguments together make building diverse genomic databases one of the most important priorities in the field. Let me now describe the annotation ecosystem as an integrated whole, because the true power of variant interpretation comes not from any single tool or database, but from the systematic integration of all these resources into a coherent workflow. Think of the annotation ecosystem as a series of converging filters and enrichment steps. You start with millions of variants and progressively narrow the list to the small number most likely to be relevant, while adding layers of evidence that increase confidence in interpretation. The first layer is genomic location and functional consequence from VIP, NOVAR, or SNP-F. This layer tells you what each variant does to the gene and its product. The second layer is population frequency from DB, SNP, and GEOMAD. This layer removes variants too common to be the cause of rare disease. The third layer is clinical database lookup in ClinVar and locus-specific databases. This layer directly answers whether the variant is already known to cause disease. The fourth layer is gene disease matching through OMIM and the literature. This layer asks whether the gene harboring the variant is relevant to the patient's phenotype. The fifth layer is computation pathogenicity prediction from CAD-EVAR and alpha-missense. This layer provides probabilistic evidence about likely functional impact for variants without direct clinical documentation. The sixth layer is evolutionary evidence from phylo. When such a variant is also de novo, meaning it arose in the patient and was not inherited from either parent, the evidence becomes essentially conclusive for most Mendelian disorders. Before we close, it is important to be honest about the limitations of the annotation ecosystem and the directions in which the field is moving to address them. No review of these tools would be complete without acknowledging what they cannot yet do reliably. The most significant limitation is the interpretation of non-coding variants. The human genome is 3.2 billion base pairs, but only about 50 million of those base pairs are well-annotated exons with established frameworks for interpreting variants. The remaining 98%, including promoters, enhancers, intronic regulatory elements, and the vast intergenic expanses, is much harder to interpret. We know that pathogenic variants occur in non-coding regions. We have biochemical assays from ENCODE that identify which non-coding sequences are functional. But for any given patient's non-coding variant, distinguishing pathogenic from benign remains extremely difficult. Deep learning models trained on regulatory element activity, such as DeepSeek and Informer, are showing promise for predicting the functional effects of non-coding variants on gene expression, but clinical-grade interpretation of non-coding variants remains an unsolved problem. The second significant limitation is the interpretation of structural variants. While tools like NotSV and databases like DGV and DEFIR have improved structural variant annotation substantially, the evidence base for classifying structural variants. Looking forward, the integration of artificial intelligence into variant interpretation is accelerating. Large language models trained on the biomedical literature are being developed to assist with integrating all available evidence for a variant and generating interpretive summaries. The alpha-missense model represents a new paradigm where structural protein modeling informs pathogenicity prediction. The field is moving rapidly, and the annotation tools available in 5 years will almost certainly be substantially more powerful than those available today. Let us close Chapter 7 by drawing together the key concepts and practical lessons from this lecture into a framework you can apply immediately. The first and most fundamental concept is that sequencing generates data, not diagnosis. A raw list of millions of variants has no clinical meaning until it is systematically annotated and filtered. Annotation adds meaning through 4 questions: location, functional consequence, population frequency, and clinical significance. The 3-tier database system is the backbone of clinical variant interpretation. Tier 1, DBSNP, catalogs known variants and assigns RS numbers. Tier 2, NOMADE, provides allele frequencies across diverse ancestries from over 140,000 individuals, enabling powerful filters based on population frequency. Tier 3, the ClinVar, provides clinically curated classifications of variants with evidence-based pathogenicity assessments. Multiple computational tools complement each other. VAP, ANNOVAR, and SNP-F each annotate location and consequence with different emphasis. CADI, REVEAL, and alpha-missense each predict pathogenicity through different approaches and should be used in combination. No single tool is sufficient, and convergence across multiple tools increases confidence. Population diversity in databases is not optional. Accurate variant interpretation for patients of any ancestry requires population match reference data. The ongoing expansion of the ACMGM framework provides a systematic approach to integrating multiple lines of evidence into a defensible and consistent classification. The clinical genomics workflow integrates all of these tools and databases into a systematic pipeline that can convert 25,000 exome variants into a single confirmed diagnosis, transforming a patient's diagnostic odyssey into a clear molecular answer that guides treatment and family counseling. I want to close this chapter with a broader reflection on why the annotation infrastructure we have discussed is so important beyond the technical details. The reason we care about variant annotation is ultimately about patients. Every year, hundreds of thousands of patients and families with rare genetic diseases undergo sequencing, hoping to finally understand what is wrong and why. Many of them have been through years of inconclusive tests, multiple specialists, and the profound anxiety of not knowing variant annotation is what converts the raw output of a sequencing machine into the answer that ends the diagnostic odyssey. When annotation works well, and it increasingly does for well-studied genes and well-represented populations, it is transformative. A de novo SCM1, a variant annotated with CAT32, absent from NOMAD, classified pathogenic in ClinVar in a child with early onset seizures and developmental delay, is a diagnosis. It is Dravet syndrome. The diagnosis changes everything: the treatment selection, the prognosis, the genetic counseling, the family's understanding of what happened and why. But the power of annotation is unequally distributed. Patients from populations well represented in NOMAD and ClinVar benefit from a richer evidence base. Patients from underrepresented populations are more likely to receive VUS results, less likely to receive definitive diagnoses, and more likely to be harmed by. The work now is to ensure that infrastructure serves all patients equally, regardless of where they or their ancestors came from. That is the next frontier, and it is as much a social and ethical challenge as a scientific one. I will see you in the next chapter.